For this experiment, we'll be undergoing the green reduction of 5-hydroxymethylfurfural to 2,5-bis hydroxymethylfuran. Before we go any further, I'd like to go over the topic of green chemistry, which is the design of chemical products and reactions that reduce and eliminate the use and generation of hazardous substances. So of the 12 main principles of green chemistry, we'll be using three of them throughout this experiment. The first one is number 5, which is the use of green and renewable solvents such as ethanol due to its high abundance in nature and its safe to use. Number 7 is the use of green reactions derived from its biomass, such as 5-hydroxymethylfurfural, the primary reactant in this experiment, derived from the cellulose. And then number 10, which is the degradation of byproducts into harmless substances, such as converting ethanol into sodium ethoxide at the end of the experiment. So a little bit of background information into 5-hydroxymethylfurfural, or HMF. It is an organic compound made from dehydrating carbohydrates like cellulose in the presence of an acid catalyst. It can also occur naturally in many heated and processed foods containing sugars after heating it in the oven. And it is very reactive because of the two functional groups it has, which is a primary alcohol shown here on the left of the structure and the aldehyde shown to the right. The other product that we're going to be making in this experiment is the 2,5-bis hydroxymethylfuran, or BHMF, showing two primary alcohols on either end of the furan ring, making it a symmetric structure, and also a little more stable than that of the HMF. It's made synthetically through selective hydrogenation of HMF, which in this case is converting the aldehyde as shown to the right of HMF, which is a carbonyl attached to a hydrogen, to primary alcohol, in which it breaks up the carbonyl and causes the oxygen to be protonated. This structure is mainly used as a building block for polyesters and polyurethanes, and is a versatile intermediate for the production of biofuels as an alternative to gasoline. This is the reduction mechanism of HMF into BHMF using sodium borohydride in the solvent ethanol. The reaction has two specific steps, the first one being the nucleophilic addition step, in which the sodium borohydride interacts with the carbonyl by having one of the hydrides being removed from the sodium borohydride and binding directly to the carbonyl, causing the pi bond to break and go directly to the oxygen. This causes the anion on the oxygen, and to stabilize it, the leftover borohydride interacts with the oxygen to form a borate complex, and then the sodium interacts with the borate complex in order to maintain charge neutrality. This forms the intermediate that is shown in the middle of the reaction step. The next step is the proton transfer reaction, in which you would add a certain amount of water and hydrochloric acid in order to quench the reaction. The hydrochloric acid is present mainly to apply an additional amount of hydrogen ions and to make the water more acidic in order to interact with the oxygen, and then the oxygen will then be protonated and kick off the BH3 in the form of borane, as well as having the sodium interact with the hydroxide that is to form sodium hydroxide. So now you have two byproducts, sodium hydroxide and BH3 formed, along with your final product, that being a primary alcohol, which is referencing to that being of the BHMF product. Before we go any further, I'd like to point out the hazards in sodium borohydride. It is very water reactive, meaning that any contact with water can cause flammable hydrogen gas to release and ignite spontaneously, which is why we are using the solvent ethanol instead of water in this experiment. So while you are cleaning up, make sure that you dispose of the chemical contaminants in the appropriate waste jugs. They're going to be labeled aqueous and organic waste. The aqueous waste should be any water contaminated substances, and then the organic waste will be that of the ethanol and the sodium borohydride used in the experiment. Any solids sodium borohydride left over in the experiment should be put in the solid waste bucket as well. And whenever you are using the sodium borohydride, make sure that you cap the sodium borohydride after use to prevent any water exposure to the sodium borohydride. So now we can go over the experimental procedure for the green reduction of HMF to BHMF. First, you're going to add 1 gram of HMF into a 50 milliliter round-bound flask and dissolve in 8 milliliters of absolute ethanol. Then, spout the starting material, the HMF solution, onto a CLC plate using a capillary tube. Then place the flask in an ice bath to get the temperature down to 0 degrees Celsius, and then once it reaches the desired temperature, you're going to stir the mixture with the stir bar for about 15 minutes. And then add 0.18 grams of sodium borohydride into the flask while it mixes. Be sure to remember that the sodium borohydride absorbs water very easily from the atmosphere, so it's best to add the sodium borohydride very quickly. After the reaction mixed for about 10 minutes, you're going to spot the reaction mixture onto the same TLC plate next to the starting material. Once the spot dries, you're going to develop the plate in a 70% ethyl acetate and hexane mixture. What this means is if you had 100 milliliters of this mixture, 70 milliliters of that is going to be ethyl acetate, and the other 30 milliliters is going to be that of hexane. Then you're going to mark the spots under UV light and develop it in the KMnO4 solution. Note that if the reaction was not completed and the starting material is still present on the TLC plate, you're going to add another 0.18 grams of sodium borohydride to the mixture and stir for another 10 minutes at 0 degrees Celsius. So in case any of you are confused about what we mean for the TLC plate, the starting material is going to be the HMF in the ethanol before the reaction takes place. That is going to be at sample spot A on the TLC plate shown here in the diagram. The sample spot B is the mixture after the reaction, which should have the BHMF solution. And that's going to be placed to the right of sample spot A with the starting material. Then you're going to develop that with the mobile phase, and then both samples should look different from each other. So now we can go over the workup and the analysis of the product BHMF. So first, you're going to add a couple drops of 2 molar HCl to quench the reaction mixture after TLC analysis. Quenching in this scenario is to react with any remaining sodium borohydride left over in the solution, as well as any unreacted HMF. Then you're going to add the ethyl acetate to the mixture and extract the organic layer into another flask using a separatory funnel. So after adding the ethyl acetate to the water and the solution, you're going to pour it into the separatory funnel, mix it vigorously, and then wait for the layers to separate. You're going to have the organic layer on top, which contains the ethyl acetate with the product, and the aqueous layer, which contains the water. Then you're going to drain the water away from the organic layer, and then put the organic layer into a separate beaker, and then place the aqueous layer back into the separatory funnel. You're going to add more ethyl acetate to the aqueous layer to extract any remaining organic material that could have been trapped in the water. Upon separation, you're going to combine the 2 organic layers they collected and dry the solution with the sodium sulfate for 15 minutes. After the 15 minutes are up, you're going to filter the solution using a glass funnel with cotton or filter paper into a pre-weighed 100 milliliter round bond flask. And after all of the filtration has stopped, you're going to rinse the funnel with an additional 3 milliliters of ethyl acetate to collect any remaining product that could be trapped within the sodium sulfate. You're going to use the rotary evaporator to obtain the yellow solid by evaporating all of the ethyl acetate from the flask. The way it works is that there will be a vacuum placed in the chamber connected to the round bond flask and cause the ethyl acetate to boil well below its boiling point. Then the ethyl acetate will be collected from the condenser and cause it to cool down and drip into the collection flask. So all that should be left over in your round bond flask should be the yellow product. You're going to weigh the flask to get a yield for the reaction and obtain the melting point range of the dry BHMF product. Note that the TA will help you in using the rotary evaporator in the experiment.